Hi to all of you. Welcome to the third lecture of the biosignaling series of lectures. In the current lecture, we'll be discussing the G protein signaling of hormones of class 2 hormones particularly. In this lecture, we will be focusing on the G protein coupled receptors and how they transduce the signal to the internal conveyances of the cell, which includes the G protein activation. And we'll be discussing G proteins, their types, and the cascading leading to the response to the hormones binded to the surface receptor on the cell. What are G protein coupled receptors? They are specialized transmembrane receptors present on the surface of a target cell which respond to the hydrophilic class 2 types hormones. These are the receptors named because of they interact with the G proteins which are located within the cytosol of the cell, located internal to the plasma membrane. And they are also referred to as serpentine receptors because they mimic the structure of a snake in having seven different transmembrane helices located well within the plasma membrane. They are the largest known receptor families, and usually the genes which code these proteins and these receptors account about 1% of the human genome, and they comprise receptors for a diverse array of molecules like neurotransmitters, odorants, lipids, neuropeptides, and large glycoprotein hormones. The structure and function of the GPCRs were studied first of all by Lifkowitz and Kobilka, who later on won the Nobel Prize for 2012 for their discovery in studying the structure and function of these GPCRs in response to the hormones. Focusing on the structure of the GPCRs, as I already told you, they have a well distinct alpha helical structures which are making seven transmembrane domains in the plasma membrane of a target cell. Actually, the structure of a GPCR can be diverged or characterized into three distinct structures. First is the amino terminal end, which faces the outside cell to interact with the environment in which the target cell is and to respond to the ligand or a hormone to which it interacts and binds and forms a hormone receptor complex. And the distinct 7 alpha helices are the ones which transverse the plasma membrane. These helices are connected together by different lengths of the loops in between them. And then there is a carboxyl terminal end, which is the end which faces the cytosol part, and this is the end which interacts with the attached G proteins. It has a ligand binding site. Ligand binding site has three loops which are on the outer surface of the cell, as you can see here, and terminal, and then the three loops which are on the outer surface to interact with the ligand or hormone here. And then there is a docking site which is also made up of three different kinds of loops which are present on the inner surface or cytoplasmic side of the cell. These are the interaction sites which interact with the accompanying G proteins located inside the cell with the help of these loops and the structure present in the G protein in themselves, so as to activate the attached G protein. G proteins are named because they actually bound themselves to the guanine nucleotide, which functions as a prosthetic group in their structure. And since guanine nucleotide in cells is present in two different forms, GDP or GTP, the G protein itself also exists as two different forms. When the G proteins are bound to the GDP, they are in inactive form, and when they are bound to the GTP, they are in active form. So for relaying the information of a hormone to inside cell and to create a cascade and to create a second messenger, the G protein have to do the switching from GDP to GTP and get activated. This happens when hormone binds to the serpentine receptors or GPCRs. So in inactive form, when they are bound to GDP, they are present as heterotrimeric in nature, consists of three different kinds of polymers, polypeptides, which are alpha, beta, and gamma. So these interact and bring out together to form the inactive form of a G protein, which is bound to GDP. And they are held inside the cytoplasm on the inner surface of the plasma membrane with the help of a lipid chains that are covalently bound to the alpha and the gamma part of this heterotrimer. As I already told you that guanine nucleotides helps in the activation and deactivation. Actually, this guanine nucleotide is present on the G alpha subunit of this heterotrimer, and when it is present as GDP, the G protein is usually inactive, and G alpha has a higher affinity for binding with the two other subunits, that is beta and gamma, so they exist as a trimer in this condition when alpha is bound to GDP. When there exists as a GTP GDP switching, when GDP is changed or exchanged to GTP, then this alpha has a lower affinity to bind to the other two subunits beta and alpha, so they come apart and dissociate to form the G alpha GTP separate and G beta gamma separate. This helps in the activation process. Then the relay and the signal is carried downwards by this activated alpha, which is bound to GTP. So the two conformations are interconvertible via activation of the GPCRs. This causes the GDP-GTP switching on the G-alpha unit of the trimer, and each then dissociate, and G-alpha subunit in turn is then free to activate the effector protein. The effector proteins are different kinds of proteins depending upon the type of a GPCR. For example, adenylyl cyclase is one of the effector, which then relays the information of the activated alpha subunit of this G protein. Then the relay signaling is done by the another effector protein, which is also membrane bound. So what happens, the ligand binders ligand or the hormone binds to the serpentine receptors, then they relay this information to the G protein. G protein causes GDP GTP switching. When it binds to GTP, alpha subunit dissociates itself from the trimer. When the dissociation happens, the alpha subunit, which is now activated, activates the effector protein. For example, here sometimes the effector protein can be adenylyl cyclase. So what we learned so far about the G proteins is that G proteins act as mediators between the serpentine receptors and the effector protein or effector enzyme. And G proteins relay the information from the serpentine receptors to the effector, and in doing so it exists in two different conformational patterns. In the on state, the G alpha is usually bound to the GTP where it is activated, and in the off stage it is bound to the GDP, and in on stage when it is bound to the GTP, we know it is existing only as a separate from the trimer. It exists as alpha GTP, which activates the effector, and when it has to go and get converted back to the trimer or inactive form, it has to hydrolyze the GTP bound to it and get converted back to inactive state. This hydrolysis is the intrinsic GTPase activity of this G protein. So G proteins are capable of converting back to the inactive form on their own. They have the capability of hydrolyzing GTP back to GDP and get converted to an inactive trimer state. Furthermore, as G alpha is now the mediator of the information downstream in the cell, depending upon the type of a G alpha, we have different kinds of signalings, and hence we can divide the G alpha subunit into four different categories. One is the stimulatory type, which is referred to as GS, where G alpha leads to the stimulatory action, leads to the increased activity and expression of adrenal cyclase, increased expression and increased quantities of the second messenger cyclic AMP, as well as increase the cardiac calcium. GI is that G alpha which is inhibitory in action. It leads to the antagonistic effect of the stimulatory G protein, leading to the decrease in adrenal cyclase activity as well as expression, decreased expression and concentration of the cyclic AMP, as well as increase the potassium channels activity. Then there is another one which is stimulatory as well. It's called as GQ. It is the special type of an G alpha which leads to the increase in the expression of phospholipase C as well as phospholipase beta 1, which increases the second messenger IP3 and PIK3. What are these IP3 and PIK3? We will be discussing them separately in some other lecture. And then there is another fourth one of which the function is unknown, but it is known that it affects the calcium channels and leads to the increased expression of the calcium channels in the target cell. So finally, summarizing the G protein cascade mechanism is that you can see in the accompanying diagram of 7 steps. What happens is that in the first step, ligand binds or the hormone binds to the serpentine receptors on the outside, interacting with the loops and the N-terminal region of the serpentine receptors, GPCRs. It in turn forms the ligand receptor complex or hormone receptor complex, which in turn in the second step leads to the GTP switching. GTP switch the GDP leads to the breakage of this trimer into the gamma beta and alpha GTP, which is the mediator. This then when activated goes to the effector protein and in turn activates it. For example, here it is a cyclic adrenal cyclase, which leads to the production of the more and more cyclic AMP, which acts as a second messenger for relaying the information inside the cell. And later on, because of its own GTPase activity, GTP is hydrolyzed back to GDP, and this enables the alpha to have a higher affinity for the gamma and beta. It binds back and gets inactivated, and this is the cascade mechanism on the surface of the target cell in response to the binding of a ligand or a hormone. What happens afterwards is the action of a second messenger, which for example in this case is the cyclic AMP produced by the adrenal cyclase, which we will be discussing in a separate lecture. So this was the essentials of the GPCR and G protein signaling. We'll be discussing more about the signaling cascades in the series of lectures to come. Meanwhile, you can visit my Amazon page for the number of different kinds of books which I have written on biochemistry and molecular biology, which might be beneficial to you. And do subscribe to my channel for further videos which I will upload in due time.